Okay, I think we will get started here. Um, Hello, everyone, and welcome to the Enhanced Decision Making webinar series. This webinar has been made possible in part through funding under the Saskatchewan Ministry of Agriculture's ADOPT program. We're also happy to be part of the Biotechnology Week in Saskatchewan. Today, we'll look at improved bottom line with DNA Parentage Verification and genomic profiling. My name is Obion Hadurin, and I'm the Prince Albert Regional Livestock Specialist with the Ministry of Agriculture, and I'll be chairing the webinar today. Our presenters for this webinar are Michelle Miller with Delta Genomics and Kathy Larson with Western Beef Development Center. Michelle completed her Bachelor of Science in Molecular Genetics from the University of Alberta and Master of Science in Human Genetics from McGill University. Michelle has recently completed her Master's in Business Administration at the University of Alberta. In her position as the Director of Operations at Delta Genomics, Michelle is responsible for the laboratory's operations, specifically process improvement and optimization, financial management, customer service, and human resource activities. Our second speaker, Kathy Larson, has been the research economist for Western Beef Development Center for the last 5 years. She has been working to provide tools and training on cost of production, interviewing young ranchers across Saskatchewan on their management and marketing practices, and rolling out the Western Canadian cow-calf survey. Originally from a purebred cattle and grain farm near Tyvan in southeastern Saskatchewan, she now resides with her husband and children at Prince Albert, where she works from a home office. Before we begin the webinar today, I have a few housekeeping notes. This presentation is scheduled to take about 3/4 of an hour before we have some time for questions or comments. If you have questions during the presentation, you may type them in the question box in the bottom right-hand side of the control panel, and we can answer them at the end of the presentation. The question box can also be used to communicate any difficulties you may have with sound or presenter speed. During the presentation, you'll be muted, so there is no feedback that will interfere with audio. Please ensure that the volume on your speakers is turned up. And if you're having difficulty with your speakers, there is also a telephone option, which requires you to dial in to listen through a phone while viewing the slides on your computer screen. And if you're viewing the webinar today from a computer with dial-up, you may experience some delay in the audio or the slides. If we run into a delay with the webinar, please stay online and our host will reconnect. And after you exit the webinar today, a brief evaluation will appear. Please take time to fill it out. It will help us plan future events. A link to the recorded webinar will also be sent out to all registered participants and will be posted on our website following the live presentation. So at this time, I would like to hand things over to our first speaker, Michelle Miller with Delta Genomics. Michelle?
Thank you. Thank you, Obi. I'd like to thank everyone with the Western Beef Development Center for giving me the opportunity to introduce how a DNA lab really works. So as Obi mentioned, I'm with Delta Genomics, and we're located in downtown Edmonton in the Old Bay building, which is now known as Enterprise Square. So what I want to talk to you today is give you an overview of Delta Genomics and really the process that goes on in a DNA laboratory, specifically to complete SNP parentage. And then I'll mention a few of the costs and also where the genomics industry is going. So Delta Genomics, just a brief introduction, was incorporated as a not-for-profit organization in 2011. In 2014, the company spun out of the University of Alberta and is now a private not-for-profit organization. Right now we have 13 employees, and last year we processed a total of just over 42,000 samples. As of yesterday, we had processed almost 33,000 samples in 2015, so it looks like we're on our way to either meet or surpass the amount of samples we did last year. The big goal of Delta Genomics, though, is really to increase the profitability, competitiveness, and sustainability of the Canadian livestock industry. And SNP Parentage is one of the tools that we can use to help do this. But looking at the big picture, we really use our ties with Livestock Gen Tech at the University of Alberta. And we really collaborate with the researchers there in order to bring the research that's done in an academic setting into a meaningful product that we can provide for industry to help grow the profitability, competitiveness, and sustainability of the livestock industry. So now I'm going to start with a brief, I want to make sure that I know that not everyone knows what happens in a DNA laboratory, and I've heard some people even describe it as magic. What I want, if you don't get anything from this presentation today except this, please know that there is absolutely nothing magical about what we do. It is all very much a process. It is process-driven, and there are distinct steps and timelines that we take every time we receive a sample. So moving forward, there's a brief introduction to DNA. So DNA is, it looks like the picture on the left, like a spiral staircase. It's made up of units called base pairs, and there are 4 different base pairs represented by the letters A, T, G, and C. Cattle have about 2.8 billion base pairs, and as a comparison, humans have about 3 billion. So in terms of the size or the amount of DNA that we have, we're not actually that different from cattle. So a lot of people have heard the word SNP before, and what SNPs are if we look at the base pairs of an animal, we can see here that the middle bull, represented by the blue bull, has a T where the other 2 animals have Gs. So SNPs are just changes, small changes in the content of the DNA, and these are things that you may or may not see on the outside of the animal. If you look further down, we can see that the bottom animal has an A where the other 2 have Cs.
Hi, Michelle.
So tell us which.
Michelle, sorry. Before I just want to interject. The slide is still at the magic heart, so I don't know if it's changing in yours. It's not changing here, and I think I've seen some other comments that it's not being translated.
Oh, no, I haven't. I actually haven't seen any comments, but my internet is up. I can try switching the internet if that makes.
Okay, it's still showing the magic heart.
Okay.
Yeah.
So I'm going to switch my internet connection and see if that works.
Okay.
Which slide is it on now?
Oh, okay. It is on Snips. So if,
it's on Snips?
Yeah, if you will be able to, if you can go back to the magic hat and then take us through again the slide after the magic hat, that would be great.
Sure. Okay, so did you hear everything I was saying, though?
Yes, we could hear you, but we couldn't, match them up with. Yeah, hello?
Oh.
Yeah, we could hear you.
Yep, I'm here.
But we couldn't match what you were saying with the slides.
Perfect. Okay, I'll go through these ones again. So to introduce DNA, DNA really does look like a spiral staircase in the picture you can see on the left. It's represented, and DNA is made up of things called base pairs. There are 4 types of base pairs, each represented by the letter A, T, G, and C. Cattle have about 2.8 billion, as I mentioned before, and humans, in comparison, have 3 billion. So looking at what SNPs are in particular, we can see the DNA base pairs for 3 animals. And if we look at the first SNP, shown in blue, we can see that the middle animal has a T where the other 2 animals have Gs. And if we look further down, we can see that the bottom animal has an A where the other 2 have Cs. So SNPs are just small changes in the DNA content and in the base pairs. So SNPs are really a measure of DNA content. And Snips are what we can use to determine parentage assignments. So if we look at the process of a SNP lab, there are really 4 main areas. The first is sample inventory, where we go through all the samples that come into the lab, we catalog them, and record the information for each animal that we receive a sample for. Next is the DNA extraction. This is where we actually take the DNA out of the sample. The third is SNP genotyping, where we do the actual, like the really technical lab work to get the SNPs out of the DNA that we extracted. And the 4th step is data analysis, which is actually one of the trickier steps. So if we look at each step in a little bit more detail, sample inventory takes about 1 to 2 days, depending on how many samples we receive. So this involves recording all the sample information on an animal, storing those original samples, and also storing the DNA after it's been extracted in these plates that you can see on the right have a barcode on the pallet, and each one of the tubes, there are 96 in each pallet, has a 2-dimensional barcode. So that barcode gets assigned to every sample that we extract DNA on. I always get asked, what kind of samples do you guys accept? And really, we accept a wide range of samples. Starting on the left, we take semen straws, whole blood, any sort of tissue tag, like an Allflex tag or a Typifix tag. Typifix is the one shown on the far right. Typifix also makes the tag that does not leave a tag in the ear. It just collects the sample, and that's the one on the bottom there. And of course, we accept hair, either put into a paper envelope or put on a hair card. So those samples then go into DNA extraction, and this takes 2 to 3 days. The cutting and cleaning of the hair, in particular, is probably the step that takes the longest, just because our technicians go through, take each hair. If it's really dirty, they'll give it a good clean. But also, we do this one by one. So that's why it takes the longest. Then we put the samples, 96 at a time, and we do the actual DNA extraction to get the DNA out of the hair. And then again, 96 at a time, we want to find out how much DNA we have in each sample, because for the test requested, we usually have to have the same concentration of DNA across all the samples on a plate. So what we do is we normalize the samples then.
Typically, the peak happens
Step 3
about 7
is
days
the
after
SNP genotyping.
It sounds like
takes
that's sort
3
of
to 4 days.
extended over the last week or so. It's been about a week, I think, since we chatted. It was the 23rd of October, if I remember right.
OB?
Yeah,
So it's been just over a week.
something is
So that peak is probably
other people.
okay. I think yeah, someone
Okay.
joined in. So
okay, no worries. So with the third step with SNP genotyping, we actually take 2 sets of those 96 samples so that we genotype 192 animals at the same time. We then put the samples, the DNA, on something called a spectrochip, which is what we use for genotyping. And then we scan that chip in a machine, as you can see on the right, which is actually mass spectrometry. So we can determine which base pair, either the A, T, G, or C, is present in each animal based on the mass of the DNA. And then the 4th step, data analysis, is actually in 2 parts, and this takes about 1 to 2 days. So the machine shown in the previous slide generates results that look like this. And obviously, this is not something that anyone can just go look at and determine the SNP profile. Some computers and computer programs are required to translate that graph into the SNPs of each animal. So that's the first part of data analysis, where we take the raw data and make it into something that we can actually read. The second part of data analysis is when we have the SNPs of the calf that we're trying to do the parentage on, and we take the SNP profiles of the possible bulls that could have sired that calf, and we compare each of the SNPs. So we can see here that the top bull does not match the calf. The second SNP, the bottom bull doesn't match the calf. And again, on the third SNP, the bottom bull again does not match the calf. So by a process of exclusion, we determine that the middle bull is the sire of the calf. So that's basically the parentage process. Now, how much does this cost? A basic parentage test, which will give you between 95 and 118 SNPs per animal, costs $20 per animal genotyped. So what that means is if you do parentage the first year, you'll want to genotype all your bulls, as well as the calves. But if you use those same bulls the next year and you do parentage again, you don't have to re-genotype those bulls. We keep the genotype profiles on and stored in our servers indefinitely. And also, if you would like to see the parentage SNPs, we can also send you those as well. And it's basically a list of the A, T, Gs, and Cs. So moving beyond parentage, you've probably heard the word genomics used a lot. And genomic profiling is a term that's becoming much more common. And what genomic profiling does is actually generate 26 to 130,000 SNPs right now, instead of the 95 to 118 that we can use for parentage. And this is a little more expensive. It can range from about $60 to $125, depending on how many SNPs you're doing. But what this genomic profiling is where we take the SNP genotypes with these large numbers of SNPs, and we combine them with the data from your EPDs to get these genomically enhanced EPDs. And what these are is they're more accurate EPDs earlier in the animal's life. So if we were to get genomically enhanced EPDs on one of your bulls, we could have a similar accuracy when the bull is 1 or 2 years old, as opposed to when it's 5 or 6 years old and has already had progeny, and we've seen how those progeny perform. So what this does is it leads to better breeding selection for the traits of interest to you. So you can get genomically enhanced EPDs, and as you know, EPDs on traits that are of interest, whether they be carcass traits, fertility traits, and so on. Right now, the seed stock sector is really getting started on using these genomically enhanced EPDs as a tool, and we're currently starting up some projects to start bringing genomically enhanced EPDs to the commercial sector. And we're really starting to focus on RFI and also some of the carcass traits. So on behalf of the folks in the lab, as you can see here, we want to say a big thank you for your attention, and also to the Western Beef Development Center for allowing us to present today. Thank you. Obi?
Sorry, I didn't unmute myself there. I would like to remind everyone that these presentations, you can download them up from you'll see a section, a pane on the right-hand side that says handouts. So the handouts for these presentations, the two presentations, can be downloaded. So right now, I would like to switch things over to our second speaker, Kathy Larson. Kathy, please take over.
Hi, can everybody hear me?
Yes, I can hear you.
I have myself muted. I apologize for that. So I am so glad that Michelle went first and essentially laid the groundwork for my presentation to flow nicely after hers. And I'm going to speak specifically on Western beef's exposure and use of DNA parentage. So I'll go through why we parentage test at Western beef, how we sample, what we've learned over our years of parentage testing. I'll share with you some results from our 2014 calf crop. And then I'll have a couple slides demonstrating what I see as economic value to parentage testing and DNA testing. And then I'll just introduce a study that we actually have currently underway at Western Beef. So why we parentage test? We actually have a purebred Black Angus herd at Western Beef, and we utilize multi-sire breeding pastures. So in order for us to paper our replacements, it's actually required by our breed association to determine the sire of those heifers. And as many breed associations do, they have an arrangement with Delta Genomics. So we actually send our tail hair samples, and they will go through the process as Michelle has just described. And we'll send those each year when we make a replacement heifer selections, and then we go about papering them with the Angus Association. Why might you parentage test? You may be in the exact same situation as Western Beef, as a purebred producer with multi-sire breeding pastures, and it becomes a requirement in order to register any of your animals. You need to go through that parentaging process in order to paper any females. You may also be just a commercial producer, and as your herd sizes increase, you're using multi-sire breeding pastures. And if you have quite a bit of consistency in your bull battery, bulls of the same breed and coat color, it becomes pretty difficult to determine and discern who's the daddy. And so it's becoming useful. You may have questions like, those are some great looking calves. I would like to either purchase brothers of the bull who sired them or from that same operation again. And so you could go through parentage testing to figure that out. On the flip side, you may have instances, unfortunately, of dystocia due to large birth weights, and you may want to know who sired those huge calves. And so those are a couple of examples of why it can make sense to do some parentage testing on your operation. So we actually have a research technician by the name of Leah Pierce, and she takes hair samples following recommended protocols found on Delta Genomics website or on the Angus Association website, and we're pulling tail hairs right from the tip of the switch. And there's actually a nice little video. I'm just going to click here, and it's going to take me to this video on YouTube, and I'll show you here. The Angus Association just shows someone grabbing a couple tail hairs right from the bull switch and yanking, and it's as simple as that, my friends. I'll pause and I'll go back to my presentation. So we do that, and every sample that we take is stored in its own envelope and labeled. So in 2014, I had some budget to test parentage on our 2014 calf crop. So it was actually just submitted samples for our steer calves. In hindsight, you know, if I could go back and do it again, I would have submitted it on all of our female or our heifers as well. We submitted those tail hairs to Quantum Genetics in Saskatchewan. We had 18 potential sires and 181 steer calves, and that was over a span of 6 breeding fields, ranging from 50 cows to 100 cows in each breeding field, and we provided submission sheets indicating for each calf who the potential sires were. So here's a table showing breeding fields. I've had them with an alphabet letter, but also tried to color code this table as well. So breeding field A, there's just over 100 females in that field. Here's the 4 bulls that were in it for 64 days. 1 bull was at 49. The last column on the right was the purchase price, what we actually paid for that bull. You can see in breeding field B, there was just 2 bulls expected to breed 50 cows, or 25 each. Same with breeding field C. Breeding field D was 4 different bulls, although you can see this 28W was only in for 12 days. And then the next slide also has some more of our breeding fields. Breeding field E is actually our replacement heifers, 55 females in that group. And then the last one was 3 cleanup bulls used for 47 days on 120 cows that have actually also gone through fixed time AI. So on average, 63-day breeding season, cow to bull ratio of 25:1, open rate overall 8.2%, and average purchase price of those bulls is just under $4,000. So what we learned when we initially submitted the results and got them back, we were a little alarmed to find that over 1/3 of the steers had no sire match. So we submitted the 18 potential sires, and there still was calves that said, no, they're none of those sires. what we discovered was we actually incorrectly labeled one of the sire's samples. So you have to be very, very careful in how you're submitting your information. We just had a number instead of the tattoo letter, so we had to get that corrected to do some retesting of samples. On the 6AI sires, we actually just had the lab bring in DNA profiles from another lab. We didn't have a semen straw for them to extract from it. And we learned through the process that DNA extraction can be done. It's easier from a tissue sample like an ear notch than a tail hair, but I think for the most part producers would choose to do a tail hair out of convenience and the ease of storage and shipping to the lab. So what we did next to try and resolve the high number of unmatched calves was retest that mislabeled sire sample. It was labeled as 1337 instead of 133Y, and we found out that he sired 21 steer calves. So that helped clear up a number of things. Then we provided down information on 15 calves. We could use both parents to try and make a match, and we discovered 6 more calves who the sires were. And for the AI sires, we purchased semen straws on those that we could get straws on and had the lab extract DNA and create a new DNA profile, but it didn't result in any new matches. And then we also submitted samples on spare bulls. So those are bulls that, to all of our records, show that they would never have been in a breeding field, and there's actually one match. And just judging by the age of the calf, it was prior to the start of the breeding season. This bull likely got in with the cows. So after all that retesting, we were down to 11 steer calves still unmatched. So 6% of the total 181 samples submitted, and they were from these various breeding fields, 5, 4, 1, and 1 from field A, F, which is those AI cows, E, and D. So how does it look in terms of results? So just bringing that table back that I showed you a couple slides ago, you can see the number of cows in each of the breeding fields, and probably of most interest to you is column 5, the number of steers sired by each of those bulls. And then because we only did steers, I just did a simple column ×2 to get a rough estimate of how many calves they could have potentially sired. I guess that there's probably some bulls that sire more heifers than steers, so it's not so simple as to do a simple ×2, but just for this example. And you can see there's steers that are bulls that have gone above what they're expected to, siring 29, over 20 steers each. And then there's other bulls that didn't even come close. These 2 bulls, as you can see, according to the parentage testing results we received, didn't sire a single calf. And then on the second slide, again, you can see a large variation from as high as 18 steers sired down to 0 as well. The last 6, you'll see they're labeled AI. So those are the AI sires that were used. And this purchase price on the 4th column is the straw price on the semen. The parentage testing results show that there was 0 steers weaned for 3 of them. And then in red in the last column, what I have found is just based on calving date what we think those steers sired in total calves, so 11 out of 20 for Duffon color, for example. If we plot the purchase price of the bulls, you may think that price is maybe a good indicator of the quality of the bull. A general rule of thumb is that we want 25 female service per bull, and you can see that there is very few at that range or above it. It's just interesting to have it plotted out. I know age can be an indicator of serving capacity, so if I plot by age of sire, the 1 yearling bull that we had sired 1 calf, and but you can see still lots of variance for each age. From the 2-year-olds, we had some that sired none to over 20 steers. Same for the 3-year-olds, one bull that sired no calves to close to 30. So we wouldn't have known this information. We just assumed that we're putting the bulls out there, that they're all going to do equal amount of work, and it certainly shows that that's not the case. And this table, very busy looking. It should be within the handouts that you can download. It's just all of the bulls and the number of steers that they've sired. And along the bottom, the last row shows the average birth weight, average weaning weight, average weight per day of age, and total pounds of steer calf weaned. I've color-coded each of the columns. In the birth weight column, if it's red, it means it's above the average birth weight. In weaning weight and weight per day of age, if it's green, it's above the average. So because we're collecting lots of phenotypic information and then blending in the genotypic information, we can get a nice table here to start making some selections on maybe bulls that we are going to keep, look for brothers of, or possibly call as well. So why do I think it pays the parentage test? If you take into consideration what it costs for a bull to keep around each year, he's probably decreasing each year about $600 based on our average purchase price of $4,000 and years of use of 4 years. It probably costs you in the range of $800 a year for feed and past or semen testing and keeping him around. There's a chance that he's going to end up injuring himself and you're going to have a risk of loss for a total of $1,800 per year per bull. And we're expecting, you know, that general rule of thumb of 25 females, so it's costing about $72 per female to maintain a bull battery. But if that bull only sires 10 calves, the cost is approaching $200, which makes fixed-time AI look pretty attractive. And on another example, the cost of raising replacements, if we look back to what calves, heifer calves are worth last fall, to the point that they're confirmed pregnant this fall, you're close to $1,900 investment. So when you're making that level of investment at these high-priced times, it makes sense to want to choose replacements from the sires whose lineage you want to have in your operation. I know we all have probably feel that our bulls are all top-notch, but I think there's variation between our bulls that we have in our herd. Some are really, really good, and some are, you know, they're pretty good. And so you probably want the heifers from the really good cows and the really good bulls. And so parentage testing can help you identify who those are. Progeny from a sire have lasting impacts in your herd. They can have impacts ranging from a decade to even a quarter century, according to some of what I've read. And with the cost of replacements approaching $2,000, the cost of having a bull around each year around $1,800 a year, it seems like a no-brainer to make that small investment of parentage testing in the range of $12 to $20 an animal, considering this long-term investment that's at hand. So Western Beef's 3-year parentage study that I wanted to bring to light here, the principal investigator on it is our lead production scientist, Dr. Bart Lardner, and the grad student that he has on the project is Stacy Domaluski. We're working with 5 ranches across the province, including Western Beef. We have 100 females at our herd out of 350 cows. So there's a total of 1,500 cows that we're following their calves for 3 years. We're taking ear notches on the calves and tail hairs on the bulls and doing parentage verification. So I just have the last line of the slide. Our first year results, so these are 2015 calves. We ended up with 4 that had no match, so we're looking to recollect samples and get them retested. And of the 4 bulls in that breeding field, 2 sired 30 calves and 2 sired 15 calves each. So you can see there is still variance that happens, so it's nice to have that information to see who's pulling their weight. So just in closing, DNA testing, it's a valuable tool, and you can start to see why it might make sense for your operation. Record keeping is a prerequisite. You need to know who your breeding field groups are. I showed you the phenotypic information, the weights and details that we've captured that helps make those parentage testing results more fruitful in your analysis. I would suggest if you aren't buying bulls that have already been genotyped that you collect samples, tail hairs on them as soon as you can after time of purchase, and grab samples on your calves as early as possible. So remember that herd sire has a long-lasting impact in your herd, and if you're going to do the DNA testing, follow the protocols that are available through the lab's websites, and you're going to probably still end up with situations with calves with no match. At least that's been our experience. And just a suggestion, if you're looking to see how it might pay off for your own operation, how about start small and just test a few of your animals and see if you feel that there's value there for you. So with that, I'll think that we'll open it up for questions. I'm just going to revert back to Obi.
Okay. Thank you very much, Kathy. Please, if you have any questions, you could type them in the question box and we'll answer them. And I would also like to remind the attendees that the slides from the two presentations are available for download, and you can find them at the handouts pane. You can download the presentations from the handouts pane. So, Kathy. Hello, Kathy?
Yeah, I'm here.
Okay. There's a question here that says, can you provide a reference for the sire impact of 10 to 25 years?
Yeah, I'll have to, I will find it and find where I was looking online and just doing searches last week in preparation of this presentation, but I will provide that for you to share. I don't have the reference right offhand. Okay. Maybe, but I will get it for you. Okay. I'll tweet it out.
Michelle, there's a question here that says, how long do you keep bull results in the lab?
The results, so the parentage results or the Snips genotypes, we keep forever.
Okay. So yeah. Forever.
The samples we also keep indefinitely, unless we use all of the sample during testing.
Okay. Are there other species apart from cattle that you work with?
Yeah, we routinely work with swine. We've also worked with goats and sheep, and we also help researchers through universities. So we've actually worked with polar bears, fruit flies, bacteria. So we can do pretty much anything in terms of species.
Okay. Please, if you have any questions, type them into the question box. Maybe we can wait for a few more seconds for any other question. Okay, there's one here that says, how many producers do you estimate use DNA parentage testing currently in Western Canada? And this will be for Michelle or Kathy, if you can jump in.
Well, I don't know an answer to that question, but if you could probably, Michelle might be able to better answer that, especially given that she knows how many samples have been submitted to her lab. She can maybe speak from their lab's experience.
Well, anyone who is seeking registration through a breed association will be doing parentage at this point. And I'm really not sure what that number is in Western Canada. In terms of the commercial side of things, we've had a few large commercial producers come to us So the actual number of producers isn't very high yet on the commercial side for SNP parentage, but we do service a couple hundred commercial producers for tests such as corn polled and coat color, and really trait-driven genetic tests like that.
And I guess just to share, we have that 3-year study that's just underway that I mentioned with the 5 cooperating ranches across the province. Those ranches are anywhere from 500 to 1,200 cows each, and they're really keen to be in the study. So I think once we sort of get some of those results out, hopefully Western Beef can help explain the value and show the value of doing that testing, and we'll see people having some information there to make that decision for their own operation.
Okay. Michelle, there's a question here that says, you know, we know that there are thousands of SNPs in the genome. How do you select the 95 to 118 SNPs for the testing?
Yeah, so that's a really good question. Through the research that has been done really since 2009, there has been a panel of about 120 SNPs that have been established as being really good for parentage use. So we and the USDA has adopted that 120 SNP panel as sort of their go-to parentage panel. It's now ISEG accepted. So we use that panel that's been determined already through research and been validated by the USDA and the International Standard for Animal Genetics.
Okay. Do you have any problems with verifying progeny from a set of full siblings?
From a set of, I'm sorry?
From a set of broader sires or, you know, siblings. Full siblings.
Yeah, absolutely. So if the sires for all the potential bulls for one calf are very closely related, we do sometimes run into trouble where through that exclusion process I just described earlier, we will get statistical ties, and we have found that is especially true when the actual sire and the dam have both been sired by the same bull. We will actually call the grandfather as being a more statistical match than the actual sire. And in those cases, we can get around that by either genotyping the dam, or we can increase the number of SNPs. So going from, say, 95 to the 26,000, for example, would give us more than enough SNPs to determine which of the closely related bulls is the true sire.
Okay. So if you still have any questions, please type them into the question box. Okay, seeing none. Please, I would like to thank Kathy and Michelle for joining us for this webinar, Improve Bottom Line with DNA Parentage and Parentage Verification and Genomic Profiling in our Enhanced Decision-Making webinar series. We also want to thank the Saskatchewan Ministry of Agriculture and the Western Beef Development Center for their support. If you have any questions regarding these topics or anything else related to agriculture, please contact your nearest Saskatchewan Agriculture Regional Office, or you can call the Agriculture Knowledge Center at 1-866-457-2377, or you can visit our website. Please take time to fill out the brief evaluation that will appear. It really helps us to plan future events. Thanks again for joining us, and have a fantastic day.
AOB,
Yep.
do you want us to just hang up here and go off?
Yeah, I'm just going to quit right now.